It's a great pleasure to be a welcomed guest of the AMS, and I always get treated very well. Yeah, one role that's very important that mycological societies can fulfill is being a resource for health and safety when it comes to mushroom poisoning concerns. So ever since we founded the Vancouver Mycological Society, I've been the point person for identifying mushrooms involved in potential poisonings. And it's a very fulfilling role. Now, I must say that most of the cases that we have are simply children or pets being exposed to mushrooms in grazing behavior or just incidental activities, and parents or caretakers get all freaked out because the little kiddo is sitting there with a mushroom in its mouth, gurgling away on the lawn or something. So about 3/4 of the cases we have are incidental exposures that lead to no problems at all. What I've done here is I've compiled the 8 cases, I think it's 8 now, that we've had so far in 2025 that are of interest to me, either because they were symptomatic or because they were known poisonous mushrooms, even if they didn't cause symptoms. So, and each year we have 100 to 150 or more calls to poison control to do with mushrooms, most of them these incidental sort of things. And there's the Poison Control Center that has a toll-free line, a local line, email contact. But if you have a suspected exposure, if there's a possible mushroom poisoning underway, there's in Alberta and Saskatchewan, it's called PADIS, Poison and Drug Information Service. And I'll give you at the end of this, I've got the information for non-BC. But this is where we get the calls. And with digital photography now, everybody's walking around with little phones and they can take pictures and they just can send them electronically. It's a blessing because we used to have to actually get hold of the mushrooms, or rarely somebody might have a film photography ability, and then you'd have to wait for the film to be developed before you'd see the suspect. But now this is really, and I can sometimes respond within minutes of getting a query. And so these are the cases. Oh, no, this is going to be an experiment here. Uh-oh. Which button is which? Right? Okay, now, most of our symptomatic cases are edible mushrooms. And this is an example. And this started off the year, February 16, location not specified. Individual ate black oyster mushrooms that a friend had grown and fried in oil with kimchi. And shortly after eating them, they had severe vomiting, felt very fatigued and weak. They look kind of skanky. There's sort of a discoloration. And quite often when people cultivate mushrooms, it's an unhygienic situation. They might have harvested them several days before they were eaten. And so this is very typical of stuff. And mushrooms must be well cooked. And many of the edible mushrooms, including pleurotus, contain proteins that are hemolytic, that disrupt the blood. But these hemolytic proteins are very heat sensitive. They break down readily in cooking. But many of the known edible mushrooms, including common button mushroom, contain these hemolytic proteins. You should always cook mushrooms, also for hygiene purposes. Case 2, Saanich, BC. An adult male ate 2 of these mushrooms because he thought it was psilocybin. And this is a gomphidius. It was very easy to identify as a gomphidius because it's yellow at the base and has decurrent gills. And you can tell from the shininess of the stem and stuff stuck of the cap that it was viscid when it was moist. And the comment that we got from the facility was that he was already high. So he apparently ate these for some reason, thinking that they were magic mushrooms. So,
Eat them raw?
yes, yeah.
Must have been desperate.
Well, sometimes in these cases, motive is very hard to establish. Now, this I found interesting. A family of several individuals ate these morels that they'd gotten. And they identified them as morels. They ate them for lunch one day, and then the following day, breakfast. And they thought that they were true morels. And then one of them got curious, and I get this happens quite often. People pick mushrooms, they eat them, and then they go online and they'll submit their photo to some app, or they start looking up the mushrooms after they've eaten the mushrooms. So you should always do that research first before eating the mushroom. And so, but all of the family members stayed asymptomatic, but they realized when they had done their little bit of postgrandial research that there wasn't actually the true morel. And the verpa, or early morels, are known to be poisonous for some people. Back east, they cause lots of problems. We've had quite a few people eat them on the west coast, and without problems, but they are, last year we had a farmer's markup that was selling verpas as true morels, and we had a symptomatic case from that of mild GI symptoms. June 15, morels. We had a person that ate mushrooms that were given to them by a friend, cooked them in butter. This couple ate them. And after about 1 hour, one of them had intermittent cramping, about 20 to 30 minute intervals, plus some dizziness when they were cramping. It was apparently quite severe cramping. And then after, the other individual remained asymptomatic. And something which is very suggestive here, and apparently this went on for several hours and it sort of passed, but something when you hear about intermittent cramping 20 to 30 minutes, that immediately will cause concerns about obstruction of the bowels, because that intermittency is what you happen if you have a bezoar or some obstruction of the bowel. You get these sort of things. So it may be that this person had a very coarse diet, got stuck up a bit, and then eventually it released and they were better. But morels must always be very well cooked. We have every year cases of morel poisoning. This was a 3-year-old who was browsing, ate some amount of this mushroom that was growing in the urban setting. This is Duncan on Vancouver Island. And the child appeared to remain asymptomatic, but the parents sent the photo of what was eaten to me, and I was able to identify it as Amanita pantherinoides, which is a fairly common spring mushroom. It's known to be toxic. It has the neurotoxins, ibotenic acid and muscimol, and it can be identified by the ring on the stem. The base of the stem has a bulb when it's intact with a little collar of veil tissue, and the edge of the cap has little grooves in it, and that section Amanita of Amanita has a sulcate or groovy margin, and so that we knew. And the color, the brown color of the cap, that's typical pantherinoides, known toxic. But with children grazing, they often eat just a small amount, and the parent isn't even sure if they actually swallowed it. They saw the child with some of it in its mouth and the broken up mushrooms. So Summerland, July, a 76-year-old male ate one of these raw on a hike. And then he thought later, oh, maybe that wasn't so good, so he had taken it home and photographed it. And I was able to establish that it was Amanita muscaria. And fortunately, he thought it was a puffball, and then he realized his error later. But when he cut open the button, he exposed a little fine line of pigment just under the cap surface, which is typical of Amanita muscaria. All the color forms, white, yellow, brown, of Amanita muscaria have this little orange pigment layer immediately under the cap surface. This individual remained asymptomatic.
Do you need a microscope to discover the layer that you were just referring to?
No, no, it's visible. In fact, I don't know if you could see it in that picture. If you were closer, yeah, it's easy to. Now this was very interesting. Fort St. John, July 29. Two young men were on a hike and they ate some of these raw. They photographed them in the field here, and I was unable to get specimen material of this, but they photographed it fortunately. And I had to use a few clues because I couldn't ask them directly questions. So there isn't an indicator of size here. So I don't know the scale, but I can guess from the lichen and the bark and the fact that it was sort of cut off at the base. And all I can imagine is that some form of tricholoma, and the stinky tricholomas are suspected to be poisonous. So that's what I think in all characters. It matches one of those things like tricholoma inaminum that have a real stinky smell like coal gas. And so they had ate them raw, evidently. And when they developed nausea and vomiting, multiple episodes. One of them had slightly elevated liver enzymes, the ALT-219, but they're otherwise healthy 20-year-old. And these liver enzymes often happen when you have vomiting, etc. Dehydration and such can throw off. So sometimes you want to test the liver enzymes a few times to make sure it's just not a temporary thing. Some mushroom poisonings cause severe liver complications.
Given that smelly, do you think they would want to eat it?
Yeah, I just, you don't want to eat raw mushrooms, you don't, yeah, it just, yeah. People are hard to explain sometimes. Often we have a lot of these puzzling cases involve people in their 20s, late teens, 20s, when they're still immortal, you know. It's too bad that immortality only lasts for a short time, yeah.
Transient. From that photo, it's a tricholoma. Like, what's the indicator?
Oh, I don't. I can only go by the visual clues. But knowing, because one of the reasons I do a lot of consulting on this is because I have vast experience in this sort of trying to identify things with limited clues. So you just see enough of these things, and because I do a lot of these forays, I've seen so many mushrooms on tables that it really narrows down the possible suspects. And then the fact that it was symptomatic, I would immediately sort of dismiss any, but there's a lot that I'm looking for groups of mushrooms that are likely to cause illness. So the tricholomas, the stinky tricholomas immediately came to mind, and the color of the gills, and the because that stinky trich group have discolored sort of off-white gills. And I was able to zoom in on it's also handy. It's really nice when people have a bit of white card or something in the picture because I can adjust the color. Sometimes these photos are pretty hard to be sure about color, etc. But anyway, I can only suppose that that's what this is. Because I only have the photo, I can only make the best guess. But fortunately, in this case, it's not too critical because it was only mild symptoms and it resolved. And this is another case where some people foraged and ate what she thought was oyster mushroom and had GI effects, gastrointestinal, and diarrhea especially, starting 5 or more hours from the time that they ate them. And the blood work was fine. Treatment with the anti-nausea, the anti-diarrhea Imodium, and some IV fluids relieved the symptoms. And this, I suspect, was from this photo, and there wasn't remaining food to be examined, but I could only suppose that she thought it was oyster mushroom, and there's things that look like oyster mushrooms. But I imagine that because there was a delay of 5 or more hours, I suspect bacterial contamination might have been an issue here. So that's about the time it would take for bacteria to get working and cause diarrhea and then resolved. And then August 20th, a young child ate some mushrooms, and these appear to be Clitocybe dealbata, which are a known muscarine-containing poisonous species that grows on lawns and in the summer warm season. And the child remained asymptomatic, and we have every year multiple cases of young children browsing on known poisonous muscarine-containing inosebes especially, and sometimes Clitocybe, and we've never had a symptomatic case of a child that's eaten these things, but we have had dog fatalities from this type of poisoning, and multiple times we've had dogs die from it. And the main symptoms of muscarine poisoning that manifest are profuse sweating, salivation, and tear production, what I call drool, sweat, and tears. And this, dogs do not perspire. They pant to cool down. So I think there's some mechanism that human may be able to cope with the effect of this type of toxin through the sweating process, which isn't available to dogs. So it could be that that's why humans don't seem to be seriously affected by these things. And so this child remained asymptomatic. And that's it for the poison cases so far in 2025. And those are just a few cases of interest. You notice that more than half of these involved edible mushrooms.
Yeah.
Yeah. So that's something. Oh, now. So now what I want to do here is ask. Oh. Shucks. There should have been a slide at the end with the PADIS, the phone numbers for the Alberta Poison Center. Anyway, it's called Poison and Drug Information Center, and it's out of Calgary and Edmonton, and both of them, I think, have non-toll, like local numbers, and then there's a toll-free provincial number, and the same PADIS system serves Saskatchewan, I think Northwest Territories. Oh yeah, there we are. Okay.
I'm going to take a picture of that.
Excellent, yeah. Yeah, if any of you want to do that, and then you can maybe share it around too if, yeah. I don't know what VoIP is, VoIP users.
Voice over Internet Protocol.
Oh, okay, great, thank you. All right. Yeah.
Got it.
Excellent. And if one of you is involved with the medical system and they probably have stickers and little things like that that could be obtained for your club and given to members. And we have stickers for the DPEC in BC that we put on our telephones of various people that might get calls have the number. Anyway, great, thank you. That's great.
Want to open?
Yeah.
You can go back up. Okay.
All right. Now, okay. I mentioned a very useful role that mycological societies can have as a resource for individuals that can help consult on suspected mushroom poisonings. Now, we had a situation where the VMS, the Vancouver Mycological Society, has turned out to serve a very useful function when we had Deathcap mushrooms introduced to North America. And this is the most deadly mushroom in the world, responsible for some 90% of mushroom fatalities around the world, poisoning fatalities. There's many more fatalities from people getting lost or getting into accidents looking for mushrooms, but when it comes to poisoning. Ah. Okay, there's a fairly complex toxin in the Amanita phalloides that takes a long time, hours and hours before you get the first symptoms. The toxins are absorbed by the time you get the first symptoms, so it can't be eliminated from the system just from cleansing the gastrointestinal system. So that's one of the reasons it's so serious. It damages the liver, and it is a European species, grows under usually non-native trees, in this case sweet chestnut. And it's a distinctive looking mushroom. You should learn to identify the genus Amanita and get to know the prominent Amanitas of your area. And we just discovered probably 2 new species to Alberta at this foray. And so there's diversity out there that we're not aware of. And okay, the toxins are called cyclopeptides, they're called amatoxins. They're these cyclic nucleic acid rings that disintegrate. They're absorbed into the blood, they break apart and then reassemble to become dangerous to the liver after, so that's why you have a delay of many hours before the symptoms begin. There's several types of mushrooms, including little brown canosebes and gallerinas that contain these toxins, small Lepiota species. But the one that causes most of the problems is the Deathcap mushroom, Amanita phalloides, 6 to 12 hours or up to 36 hours before the first symptoms. You get a period of 24 hours or more of very severe gastrointestinal cramping pain, abdominal pain, vomiting, diarrhea, rice water, cholera-like profuse diarrhea. And then you get a false remission that lasts about a day, and then about 72 hours post-ingestion, a second wave of symptoms come on, and that's when the liver is already advanced and being damaged. Yeah.
Paul, could this happen to those tiny little gallerinas?
Yeah. Yeah.
They have to have a certain quantity, surely.
That's right, yeah. And that's why they're not implicated in poisonings very often. And usually the gallerinas are where young people or inexperienced people think they might be magic mushrooms, psilocybe. So that's the only scenario where people tend to eat the little brown mushrooms. So we don't have, there's been very few little brown mushroom amatoxin poisonings, and it's the Amanita phalloides that by far is the most frequent. And so then usually death occurs about a week after the ingestion. And the most famous now case of Deathcap mushroom poisoning is Australia. Erin Patterson poisoning her family with beef Wellington. And if you go on YouTube, you can find all sorts of stuff now explaining how that came about and what she did and et cetera. But anyway, it's very serious. The Amanitas develop from a universal veil, then there's a partial veil inside, so it leaves a distinctive cup at the base called the death cup. And sometimes patches of veil material are left on the cap surface. And then the partial veil is a flap that protects the gills and keeps them nice and moist and such as the spores ripen. So that leaves the ring on the stem. Those are the characteristics you want to look for the genus Amanita. The color of Amanita phalloides is generally somehow greenish. It can range from almost white to almost black and all through browns, but there's often an olivaceous or greenish component to it. And when they're in dryish conditions, they get a satiny sheen, which is quite distinctive once you've seen them. And the stem tends to have a bit of the cap color below the veil, below the ring. And but the cup is usually pure white, and then a bit of greenish pigment on the stipe. And once you've seen them, it's a pretty easy one to recognize. The leaves here are hornbeam trees, and that's the species of tree not native to Canada that we find them under mostly in Vancouver, and I'll explain that soon. And this is the first location where they showed up in Vancouver. And this clump, the first time they fruit in a new location, they tend to produce lots and lots of fruiting bodies. And they're often mistaken for puffballs, and that's why you want to cut potential puffballs in half and make sure it's not a developing mushroom. And here's some showing some of the variety in color. And this is how I can make them float in the air. What I do is I rub my beard with a balloon and build up a static charge, and then the dried mushrooms float in the air. And so this is examples of what they look like in the field. And there you can see the color on the stem, a bit of the cap color below the ring is evident. The underside of the partial veil has a bit of the greenish color, and then it contrasts with the white vulva. So it's a very distinctive looking thing. This is my backyard in Vancouver, by the way, sweet chestnut. And my uncle.
To whom does the...
That was my uncle's skull.
Pardon me?
That was my uncle's skull.
Your uncle's skull?
Yeah. He went to U of A in 1951 and took pre-med. And he somehow inherited a skull. And they are all mycorrhizal. All amanitas our amanitas are ectomycorrhizal. They have to grow with trees. They need the tree. They're beneficial for the tree. And they're actually attached. The mycelium is attached to the rootlets of the tree, and they exchange nutrient, support each other. And this is what the mycorrhizae look like on the tree root tips. There's a little sheath, and it's actually surrounding the rootlet. And
Well, is the, not like you're gonna eat the mycelium, but does that also have the toxin in it?
I don't know if anybody's actually dug out a bunch and extracted it to test. But we would assume that it would, but it wouldn't be of issue. I might say jumping ahead, but we were able to detect Amanita phalloides in Kelowna, BC, where it's never fruited, by digging out tree roots and looking for the DNA on the tree roots. So we know that it can be in tree roots. Okay, now in Europe, it's native to Europe. And there appear to be different types of them, different clades of them in Europe. There's Mediterranean, warmer temperature ones, and Northern European, colder climate ones. They can grow with an assortment of trees. All sorts of trees are ectomycorrhizal supporters in the Pinaceae and the Fagaceae and other tree families. And the Amanita phalloides is known from both conifers and broadleaf trees in Europe. But in North America here, it's differing in the tree host. So they have been introduced into Asia, South Africa, Southern South America, Australia, New Zealand, North America, all from presumably European origins. In Europe, I tried to drum up a map of the world showing the distribution of phalloides. And because I was wondering, could they grow in Alberta? Could they grow in Edmonton and Calgary? And I was assuming, because all the records where I could find actual locations in Europe seemed to be in areas with some moderated climate by being maritime influenced, or somewhere near the coast, and I was unable to find anything right in central Eurasia, like the real, where you'd have Alberta, like, deep freeze, and northern sort of boreal, maybe bordering on permafrost, or, where you get deep freezing for a long, very cold winter. But unfortunately, when they record them, because some of the old records didn't distinguish between Amanita phalloides and Amanita citrina, and they have the dots showing both those species without distinguishing between them. So I don't know if it's but we should assume that's potential that they could grow in Alberta cities with introduced street trees. And so far, we have on the west coast, the first record of Amanita phalloides was California 1938, Oregon 1959, Washington State 1978. First finding in BC was in 1997, and apparently in 2000 teens, there were some found in Alaska. So there's about a 2-decade interval between the jurisdictions as they're moving north.
But that might be the result of climate change.
It could be. It could be it took that much time for introduced trees contaminated with them to move up and to mature enough to produce the mushrooms. It could be, we don't know. Yeah, I think it's more the mushroom moving around rather than the climate changing. Yeah.
Has it moved east?
It as separately occurs on the East Coast. On the West Coast, they are all under broadleaf trees. All of the ones in New York State and other eastern ones are under conifers. And they have none of them, I don't think, that are recorded from broadleaf trees on the East Coast. And they're not a single...
It's going north, but not east.
Yeah, no, yeah, it appears that, well, we've got the north-south barrier of the Rocky Mountains and the Sierras, so they may not have been able to cross that barrier, but that's a question that we'll just have to wait. Now that you folks are going to be aware of it, you'll be aware what Amanita phalloides might look like, and we may get a record of it. But the East Coast, oddly enough, there's not a single record, as far as I know, that's been verified from Canada, and yet they grow within miles of the border in New York State, or on the Canadian side, kilometers of the border, you know. But anyway, so okay, so first found in 1997. The way that came about was there was a mass poisoning in 1989 in the Columbia Gorge, where some Asian people of, I think, Laotian or one of the Southeast Asian countries, were gathered foraging for sweet chestnut nuts in a grove of old sweet chestnut trees, and they found mushrooms that they thought were the paddy straw mushroom growing in amongst the chestnuts. And they ate them and had severe liver damage and needed to have liver transplants to save their lives. And the most severe of there's a group, I can't, 8 or 9 individuals were involved with that mass poisoning. So we heard about that, and then I'd found out that they were in Seattle in the Arboretum at the University of Washington, and I saw them in Seattle, and then so we told our club at a couple of meetings, we showed them pictures of Amanita phalloides and said, these are now in Washington, could everybody keep an eye out for it? And then just a few years later, in 1997, a member of the Italian-Canadian community was foraging for chestnuts in an old chestnut grove in the Fraser Valley and found Amanita phalloides in amongst the sweet chestnuts. So that was the first BC record. And here's the herbarium label from the first Western North America, November 2, 1938, Del Monte Hotel in Del Monte, California, and under some ornamental tree, under shrubs and sandy soil. This is the first one ever found in Canada. This is the 1997 specimen found by Marco Pellizzari. And this was very good because he was familiar with them from the old country, and then he knew a member of the VMS who he gave them to, and then that gave them to another member of the VMS. He gave them to another member, and eventually it got to me. I was able to confirm, yes, this is definitely Amanita phalloides, because I was the only one in the VMS that had actually seen them growing in Seattle. So on that was the record. It was in Lake Eric near Mission in the Fraser Valley. And so we found them in 1998 in Victoria at Government House, the official residence of the lieutenant general or whatever it's called, representative of the crown. And under beech trees that were likely planted in 1906 when the government house was founded. For over a decade, we found them under oak, linden, and hornbeam trees that were associated with plantains and developments that were able to date to 1912 and 1925. We found them in other municipalities under sweet chestnut, hazelnuts. 2008, found the first ones in Vancouver, which I showed you pictures of earlier, under the hornbeam trees, the Carpinus betulus street tree. By 2013, we had 7 locations, or meaning trees that are reproducing them, and 6 of those trees were hornbeams. Only 1 was oak. 2014, we found them on Galliano Island. So now we got Vancouver Island, Vancouver Mainland, Fraser Valley, one of the Goofy Islands with hazelnut. 2015 was found with native Gary oak tree, the only native oak that we have in BC. And there was a native tree, so it can jump to native trees, which we're worried about, because that's when they can become invasive, is when they get out of human-created environment into the wild. And we had 3 poisonings so far, and 1 fatality caused by BC Amanita phalloides. We had another fatality that happened in Vancouver, but the man had been visiting California and ate them in California, then came back up, got sick, and died here, but that was California, yeah. Mushroom.
Paul, could you explain the verb jump, to jump? How do they?
Well, it's figurative.
Yeah, I know, but
They're located near hot springs.
what's
Yeah.
the mechanism?
What?
It transporting the
Well, the spores drift and then the mycelium begins to grow with a new tree.
Oh,
It's
it's not
a
like-
figurative thing just to say that it's moved to a native species rather than an introduced species of tree. Yeah.
I'm just curious, I know that once it's fruited, it's already starting to spread, but do they do anything with the trees that it came on, like when they find it associated with specific trees, do they do anything with it? When they find it on a specific tree, especially like an introduced tree, do they do anything with that tree? Like I know.
Oh,
once it's
no.
fruited-
At one time, people had suggested,
Repeat the question so that everybody.
yeah, she's asking if they do some treatment to try to deal with them when they find the trees producing them. And they at one time were talking about cutting down trees that they find are hosting them. But they've been in many areas where I'll go into a bit of the demographic or the epidemiology of it soon, but the only thing that we've discovered that might be a promising thing is that it appears that they stop fruiting if you fertilize with nitrogen around the tree. So it's thought that we could perhaps just start a program of applying nitrogen fertilizer in areas that are producing a lot of them. But you get a double sort of problem there, because you might increase the vitality of the tree, cause it to grow a lot faster. And then when you stop fertilizing, they might produce even more mushrooms than they did in the first place. So we don't know. But yeah, it's interesting because it's being proposed that what we do recommend is that if people see that they've got Amanita phalloides under street trees or in their yard, that we ask them to remove them whenever they see them, put them into the green city compost system or into the regular garbage, and not mow them or stomp them or kick them around to spread them. And we hope that simply by removing them, they'll get them out of the way of children and pets and people. So it'll minimize the chance of them being involved in exposure to humans. And okay, so these are the known host trees in BC. And we've got, all of them are non-native trees except for the Quercus Garryana is the native Gary oak. So that's the one that it's jumped to a native tree, and all the rest are introduced trees, mostly street trees or parks. And these are all broadleaf. And here is around the world the sort of trees that they've been recorded from, especially in Europe. And I highlighted in yellow bold the tree genera that we also have occurring natively in BC. So in Europe, they can grow as birch trees, and we've now found them in European birch in Victoria. But we have native birches, so they could conceivably go invasive on those. We have native corylus or filbert hazelnut. We have the native oak that they've already jumped to. We have spruce, pine, we have poplars, they're native. So theoretically, and suga, the hemlocks. So these are all native trees that they could potentially spread to. So it's of concern. This is just a view of one of the sites of a poisoning. 2003, a Victoria man picked and ate what he thought were puffballs. They were in the front yard. They were under Quercus robur, an English oak that is the red leaf variety of English oak. And he was into alternative medicine and wild foods, a real enthusiast. And he just, oh, I really know my mushrooms. These were puffballs. These were puffballs. Oh, no, I didn't make a mistake. So one of the mycologists from the Pacific Forestry Center visited his home and found mature ones growing in his front yard. They'd opened up and they no longer looked like puffballs. So that's why you want to cut a potential puffball open to make sure it's not a Amanita button. And he was able to be treated and he survived.
How could you tell it's not a puffball?
You slice it in half and you can see the structure of the mushroom inside.
See the cap structure there?
And the
On the right
Deathcap
hand
has
side.
no peculiarities?
Yeah, it's deadly.
No, but I mean the cut section, does it?
Well, yeah, you can see the mushroom inside it. You see the picture there.
See the cap?
Yeah, but won't a puffball look like that too?
No.
No. Puffball is solid white through.
Oh, it's solid white.
Yeah. You haven't looked at a cut open puffball?
Yes, I have.
Okay, wow. Yeah.
Okay.
See? Yeah.
Like a marshmallow.
Yeah. Okay. In 2008, a 68-year-old Korean woman ate what she thought were straw mushrooms. And they were picked on a friend's property at this junction of 200th Street and 16th Avenue. And there's a little gizmo marking that. This is interesting because over on the right side, upper top of the frame, there's some fields there. Those are the city street tree nursery, where they plant out nursery stock to harden it out before it goes from the container into the field hardening for 5 years, and then they're dug out and taken to the streets where the trees are planted. And we think that this is how they've been getting around. And this is a straw mushroom that is popular cultivated mushroom in Asia, hot temperature type mushroom. And these were actually straw mushrooms growing in big compost heaps in Stanley Park in Vancouver and UBC Botanical Gardens. And they escaped from cultivation trials at UBC into compost systems and lasted a few years outdoors in these hot compost piles. And so but anyway, so she mistook the Amanita phalloides buttons for straw mushroom. This is a very typical scenario. Many of the poisonings are Asian people mistaking them for the straw mushroom because of the similarity. Oh.
Suspect that the mycelium travel that far underground?
No. No, it's thought that because for decades they've been growing street trees and stuff in these tree farms, that they would have fruited and then the spores would have drifted to nearby areas and established. So, yeah.
Earlier you said to, if you find any, to put them in the compost. Is that what you said? How did this?
Yeah, that's what we recommend people do, to remove them, to get them so they won't be able to spread spores, but not home compost, the municipal green bins, if you have them.
And what will they do differently that makes the spores not grow?
Oh, the hot compost. I mean, they put chicken, animal remains in there, and they compost to high enough temperature to kill everything off, whereas the home compost would probably not be adequate. And
So what is the temperature that it has to be?
what?
What is the temperature of the compost that it has to be to kill the mushrooms?
They can travel through space as spores and not die.
Yeah,
I don't know.
I can't remember offhand. There are tables you can find for thermal death temperatures for different organisms. Humans, I can't remember what the human is, but there's.
I put everything in my home compost, including like the animal remains when my partner slaughters a deer.
Oh,
So that's why, but it gets really, really, really hot. So that's why I'm like, is.
that'd probably do it. Yeah,
Okay.
yeah. It just, yeah. Okay, now, we think that these have been introduced originally in just a few tree container trees that happen to be infected with the fungus in the root system, and that they would have been moved into North America and planted out, and then these mushrooms would have fruited and the spores would have drifted off to find new trees to grow with, and that's probably how it spread around. In 2015, Shannon Birch found an Amanita phalloides growing under these Gary oak trees in Saanich, and it just happened to be right outside where she worked at the experimental research station for provincial forestry. Yeah, and we found it very interesting because this was the first case of it being with a native oak tree. We only have the one species of oak in BC, but it was a native species. And the history of the site is very interesting because this is a 1926 air photo of the location, and the white star is where the Amanita phalloides was growing. These are all commercial nursery fields. The black star is the office of Leyritz Nursery, the first nursery in Western Canada. And he was a European from Netherlands, France, Belgium, Germany, and he'd gone to different institutions to learn horticulture, and he established the Leyritz Nursery in 1889, and much of the nursery industry sprang from his involvement. And here we can see the location where the phalloides grew, and there's a tree orchard sort of field right adjacent to it. And in this is 20 years later, and the fields have been converted to some smaller herbaceous crop or some garden crop rather than tree nursery. And so and now it's housing development all through there. So we think that this thing was introduced. It probably were more mature trees that he'd brought over from Europe, or somebody had brought over from Europe, eventually produced the fruiting mushrooms that would have had spores drift into all the outplanted new stock of trees that eventually became street trees around Victoria. And so this is the mechanism by which we think these things have been spreading. Now, we found the first ones in Vancouver in 2008. By 2013, there's 7 locations, 6 hornbeam trees and 1 oak tree. So in 2014, a small group of our members formed a little committee, and we decided to do a survey, and we got the city tree records. We have a park board public access database of all the street trees, and it lists various information, what species they are, the DB8 diameter at breast height, sometimes the year of planting, but they only started putting the year of planting in 1889, unfortunately, so anything before that. And then we got we're able to find mass plantings of the different target tree species like oak, chestnut, beech, and hornbeam. And we looked up their records and we found streets that had been planted all at the same time with those species of trees. So we're able to have a plot sort of set up already made for us. We assigned different locations to different people, and they'd walk up and down looking for the Amanita phalloides. And we were targeting the hornbeam because most of them had been found under hornbeam. And so we did that. We go with tape measures, and we measure the circumference of the tree at breast height and calculate the diameter that way, compare it with the database records. Unfortunately, none of them initially were in as a planting year record in the database because they all predated 1989 when they started including the planting year in the database. The card files that they transferred to the computer system didn't include planting year. And so by the time that we'd done this for a while, we had 50 trees that we had found that had Amanita phalloides, and there were only 3 that were not hornbeam. And nowhere else have I been able to find that correlation of that many of one tree species that is the only host for them, and we have. So it's somehow that predominantly hornbeam seems to be what they started out with. And we looked at mass plantings of the oaks and the other species, and we didn't find the same abundance of stuff. And we had done collections. We had one person who was collecting DNA from them. She was unfortunately sort of a funny person. She thought for some reason she was taking little bits of stem tissue instead of little bits of gill. Would have been much more efficient just to do the gills. But we got the DNA sequences to confirm. We tried to voucher every time we'd find a new record, a new tree producing them. We tried to voucher a herbarium specimen. And now UBC has ridiculous numbers of dried Amanita phalloides in the herbarium, taking up valuable shelf space. And so we have the predominance with street trees of the hornbeam species and the cultivar fastigiata, which are the pyramid hornbeam, and they're grafted onto a rootstock, and the top is grafted of the horticultural highly branchy. It looks like a shaving brush sort of thing. They're quite an attractive urban tree. And so predominantly it's been the variety fastigiata. And the age when they've been coming in is 30 to 50 years age of the trees. And we are able to find this out by doing a bit of detective work. And so that allowed us to pinpoint areas where we could say they're highly likely that they could grow in these particular locations.
Yeah. Can the hornbeam grow in Alberta?
I think so. Yeah. Is anybody here? No. Okay. This is what hornbeam trees look like. That's the fastigiata type. They're related to birch. They have birch-like leaves, catkins.
I said, yeah, they're birch family. So there's no crossover to anything else in birch? In the Vittulaceae?
Yeah. Yeah, we have them under European birch in Victoria. They've been fruiting under birch. That poplar, they grow under poplars in Europe. Yeah. So it's possible that we'll find them under poplars in North America too. And this is, me for scale. And this is what the streets planted with the hornbeam look like. They sort of bottle brush foliage is what is quite distinctive. And they're a medium-sized tree. It takes 30 to 50 years to get the full height and crown spread of the tree. And that's when they begin to produce lots of seed. And that appears to be when the Amanita phalloides begin to appear. just when it's reached. So we've mapped out locations. And in this case, we had a couple of dings that were only DNA. So we only somehow, Vivian, who was really keen on the sequencing thing, was not so keen on preserving the actual specimen. So we have records only represented by DNA, which unfortunately, yeah.
Do they kind of predominate where they are? Do other mushrooms grow there too? So people,
Yeah, other mushrooms grow there too.
oh, so people are drinking other things.
This is we found a resource that was quite valuable, and it was a history book that had an atlas of development in Vancouver. It goes decade by decade, and it shows green is undeveloped land. So there's these green, I've got little asterisks superimposed here showing locations where we found the Deathcap. And this is my house, that's where I lived in Vancouver. That's cemetery, and the first location was that dot. The second location was up there. And this is something that's important because this is my neighborhood where these were found. And I've been there for like I began to occupy that location in 1990. So between 1990 and the 18 years that I've been in that house, I'd never seen an Amanita phalloides until 2008, and they were streets that I'd walked. So we know that the mushroom just started coming up then. And that's...
After you moved into the neighborhood.
Isn't that suspicious? Yes, yeah. And cemetery too, it all pieces together, you know. So these are 1950s, and these the green there are undeveloped. They're like waste lots, empty lots. In some cases, it was like old golf courses that have been allowed to grow back in. There's some very dark green. There's like the cemetery. Light green means parks, so other little city parks and things are in light green. And there's some very dark greens, which are still agricultural fields or little farms. So there's a few of those that were scattered around the city. But several of the dots, the asterisks, are in areas where in the 1950s they were undeveloped, like that one too. And that was an important site to demonstrate this. And then this is the 1960s. And you can see the dots that were on the undeveloped thing, where the phalloides grew, or here have been developed. And that's when the street trees were planted, when these new neighborhoods were developed. And this was the Waverly, this site was very interesting because when we found them growing there, the whole little development was done in the mid-60s. Every house and several street sort of block little area was all 1960s architecture. And the whole thing looked like a great movie location because the whole neighborhood had the whole thing was built in the 60s, all was intact. And even some of the cars that were there were old. But now it's all been torn down and redeveloped, of course. Anyway, so we were able to figure out there was the 1960s because you might recall I mentioned that no tree planting years were entered into the database that predated 1989. So that database information wasn't available. And then this is the Vancouver street tree thing, and we have a lot of prunus, cherry, and plum, maple, and which are non-ectomycorrhizal. About half the city's street trees are non-ectomycorrhizal, supporting a miscellaneous of other things. This is hornbeam, which make up 3% of the city street trees. Oaks make up 4%, linden 5%, those three, and beech 3%. So these four genera of trees are potential host trees for the Amanita phalloides, and yet vast majority of them have made up of the phalloides locations were under our hornbeam. And all sorts of other mushrooms grow, so we have Amanita phalloides there, Amanita muscaria, white variety, Amanita muscaria, Amanita gemata, We have Amanita pantherinoides, lots of Russula cerulans, very common where the phylloides grow. Boletus edulis is quite common where it grows. Sclerodermas grow with them. We have lacarias, we have Amanita muscaria, and the Chalcoporus pipperatus, the pepper bolete that always grows with muscaria. We have Paxillus involutus. These trees are wonderful ectomycorrhizal mushroom supporting trees. So the diversity of other mushrooms that they bring to the city make up for the hazard that's presented by the deathcap growing with them. And these are some of the mass plantings that we studied. And we have the number of trees in the mass planting. We designated just the areas as mass plantings. We have the number of trees, the trees without the phylloides and the trees with the phylloides, and the percent of infected trees. Almost half the trees in one mass planting produced up to this point, and we've since then had more of the trees in that mass planting producing them. So we've stopped keeping the statistics after 2015 because the project just got, I think we proved what we wanted to do. And then this just shows the areas, the mass plantings, just in a graphic form to show you. And this is the total. So of the mass plantings that we looked at comprised 352 trees, and 55 of them were producing deathcap mushrooms by 2015. So 15%, just a little over 15%.
So the risk of poisoning is going up all the time.
Oh yeah. Oh, perhaps, but we've also been public education reach out. We've now got posters every autumn when we have the first reports coming in of them fruiting. We put out a public service notice reminding people that you don't want to eat mushrooms without knowing what they are. And we were able, from the maps that I showed you, we were able to pinpoint certain locations that were from the 1960s using that atlas of the development of Vancouver. So we found these locations, these 6 locations that we were able to identify as being the mid-60s where the trees were planted. And so these are the sites that were in. Almost 20% of these total 136 trees that we know were planted in the 60s are infected with it. And
But if we know where the hornbeam trees are, then we could send a team of people around and
That's what you
Check.
said you did.
and survey, that's right. That's what I'm suggesting.
yeah.
This is a citizen science part of this. So you'll have to learn how to identify a hornbeam tree. You'll want to learn to identify the oak trees that are in your cities, and find out in your neighborhood where these trees grow, and make a point of looking under them every time you happen to be by in the autumn. See what mushrooms are under there.
Which months are the fruiting seasons?
They're autumn.
Autumn.
Yeah, so we've had, if, areas that are irrigated can produce them earlier than areas that are not irrigated. But, let's see. We now know well over 115 deathcap locations in BC, over 90 in Vancouver. This is 2016, sort of look at it. Only found in late summer and fall in BC. On June 23rd, deathcaps are found in Victoria, June 24th in Vancouver. And that was a particular year when it was wet. July 18. We found them growing under an English oak on a street right adjacent to Pacific Spirit Park, and there's oak trees that have been growing wild in Pacific Spirit Park from street garden waste that people have been dumping there. So there's now these oak trees growing in a city park. There's a wild wilderness park. that has now these potential introduced host trees. And now we know that they can grow with Gary oak because we've documented the stuff in Victoria. And in 2016, a young child died after eating deathcap mushrooms in Victoria. Yeah.
Is the aminotoxin only in the fruiting body, or is it in the mycelium as well?
It's in the fruiting body, but nobody's, as far as I know, has analyzed the mycelium. But it's sort of a non-issue anyway, because it's not, yeah. Okay, it's well established in coastal British Columbia. We have to let healthcare people know that there's potential for them to have amatoxin poisonings, because if they have patients coming in with gastrointestinal symptoms, liver complications, that might not be the first thing that comes to mind. Fortunately or unfortunately, with Aaron Patterson, Deathcap iNaturalist murder in Australia making news, probably a lot of physicians are now more aware of the liver damage from Amanita phalloides poisoning than before Aaron Patterson knocked off her family members. We do the seasonal wardings. We put out posters. We have people now really aware of children and pets and things perhaps getting into these. We're hoping that we can keep recording the locations of them. We're thinking of there's certain areas where they grow a lot where we've actually put little notices up on the street. And now we're rethinking that because it's become evident that people might also seek them out for mail purposes. July 6, 2025, or 2023, a 2-year-old toddler ate pieces of mushroom growing in a backyard. The parents had been keeping track of the deathcap mushroom. They're interested in mushrooms. They thought of joining the VMS. They're well-educated, aware people, and they immediately, when they saw that their kid had been nibbling on this mushroom, took pictures of it and took the kid to emergency, and they contacted me and sent me the photos. And I wasn't quite sure. Quite often the photos are really difficult to make sense of. This one, I knew it was an amanita from the bulb, and I could see vulva and all of that. But I said, this looks like it might be, get the mushroom to me. So they couriered the mushroom to me. Actually, it's delivered by one of the poison control people who's a neighbor. And I was able to just take one look at it. I said, yeah, that's it. I made a slide, popped it in melters, confirmed that it had amyloid spores that were the right size and shape, and contacted the hospital and said, okay, well, this is the Amanita phalloides, and they'd given the child activated charcoal, and they're able to put the child under observation and monitor, and they seem to have expelled it from the system. The parents had seen her eating them, so she hadn't absorbed the toxin. So that's a case where it turned out that it was very useful to have the information out there, and the quick response prevented any tragedy from this. And one of this location was just a few meters away from a dot on that distribution map from 2016, where there was only the DNA sequence for the dot on the map, because Vivian hadn't retained the specimen. And so
what are the treatment options if you catch it early? Is it just
yeah, the activated charcoal. There's something called Ciliabibilia milk thistle extract that's protective of the liver. There's a couple of other drugs that can block the uptake of the toxin into the liver tissue. And we had a case, a child who ate in the springtime, had eaten Cuneroomyces that looked like they might be Galerina. The hospital administered a antibiotic and an anti-rejection drug, which are shown to have some effect in blocking the uptake of the amatoxin. And so they unleashed those on the child, and then we found out it wasn't an amatoxin containing thing, so that was shut down. But there are these things, they're mostly predicated on catching it in the first wave of symptoms, and then there's the remission of a day, and then the second wave. By the time the second wave happens, the liver damage is extensive. At that point, it's support, life support, until if they can make it through the crisis of the liver damage, then the liver might slowly be able to heal enough to be functional. But transplant is another option, but not everybody's a candidate for that. If the kidneys and other organs have been damaged, or there's all sorts of other organs that can be damaged to the point where liver transplant's not survivable for the patient. And then it's down. It used to be about 90% fatality from cases of ingestion of Deathcap, and now it's well below 50% of cases that result in death. And it's usually a lot being eaten and not noticed until well long after, when the second wave of illness is progressing.
I wonder if we're able to access Amatox test kits in Canada.
Yeah, you can get it in your living room, through your door, and probably at a pharmacy. Concentrated hydrochloric acid and newsprint. And you can take a piece of a mushroom and squeeze the juice onto newsprint, let it dry, squeeze more on, and you keep building up the liquid and drying it out. And then you streak across with concentrated hydrochloric acid. And if there's amatoxin, it turns green. It's called the Mixner test, I think.
Is that useful for urine as well? Because I know amatoxins
No.
can be used on urine of people who have eaten amatoxin.
Yeah. Generally, the testing is sort of beside the point. If they'd be concerned about it, amatoxins, when the symptoms have already manifested, and they are looking at ways to test for it, but there's different tissues from organs that can be excised and tested for, but generally it's not an easy thing to. But to test the mushrooms for whether they might have amatoxin, you can use this Mixner test, which is kind of a rough. And tryptamine containing mushrooms will give a red spot that turns green, or green that turns red, something like that. Whereas the amatoxins produce a green splotch. But having a bottle of concentrated hydrochloric acid kicking around for a long time isn't recommended. Okay. And so these are the sorts of posters that we're generating to put up. And this was from NAMA, the North American Mycological Association, a dog group in the Bay Area of California produced this one, which I really like because it has Deathcap mushrooms grow in this area, which I think is a good way to phrase it. And this was aimed at dog walkers. So we've sort of stolen the idea of this. This is the one we had made up for BC here. And this has the different languages. We got somebody to do the different languages, because a lot of the poisoning cases for Deathcap are people from different parts of the world. And language barriers sometimes can be significant with these cases. So we tried to get the languages that would be most applicable to our BC scene. And here we post them up here. You know, we've got the VMS Mushroom Show poster, the warning poster for the poisonous mushrooms. You've got in Calgary and Edmonton, I recall, you've got the little free library boxes and little community posting things in parks. And these are great places when mushroom season rolls around to put up just little reminders to, do not pick and eat wild mushrooms, they can hurt you, sort of thing, which is a good club project. So here's another look at one of these little free library community posting zones. And this was an area where we had a lot of the deathcaps. And then we sat down and tried to figure out where it was likely, the climate conditions, et cetera, that we would have deathcap mushrooms in British Columbia and where we know they are. So there's Vancouver Island, and we sort of mapped out the areas that have the Gary oak and where the climate is sufficiently mild and where we were thinking high probability of them occurring. So this is the Fraser Valley up into there. And then we had the Okanagan Valley too, which has very mild winters. And it happened that Leyritz, the guy who, the pioneer of nursery industry, had started the tree fruit industry in the Okanagan when he established a plantation, a tree farm nursery operation in Kelowna. So we had a student from UBC Okanagan who did a project of doing root cores on city street trees and looking for DNA of Amanita phalloides, and he found them with hornbeam trees in a city park in Kelowna, where they have never been known to fruit yet. But we know that they're in Kelowna.
So were these young trees that hadn't yet achieved the DBH?
Yeah, they were just on the verge. They were about 30 years old. And by the way, the street, I had to move, and I moved into a new place in the same neighborhood. And the street that I'm on now had trees planted in 1996, 1995 and 1996. There were hornbeams, oaks, beeches, and planted on East 37th Avenue, right by the cemetery. And this last year, the first Amanita phalloides showed up in the hornbeam tree planted in 1995, 30 years ago, in front of the house that the past president of the Vancouver Mycological Society used to live in. So there's a strong association with mycologists. So it turns out that they're in Kelowna and could be all through there. Any place with a mild winter could host them. And we don't know if they're able to survive in the Alberta type colder parts of Alberta like Edmonton, drier parts like Calgary. But we think that,
Yeah, Paul, what's the European footprint like? Does it get into Northern Europe?
There's a problem there because I looked at Mycoportal, and they have the old records of Amanita phalloides are confused with Amanita citrina. Amanita citrina used to be called Amanita phalloides variety citrina. So when you call up the Mycoportal, and you got the Google Earth sort of thing with all the dots, all the records from all the herbaria around the world, all the dots are Amanita phalloides citrina. And I tried to just have it search and show only phalloides, but always dumps in the citrinas too. And the citrinas, I try to search only citrina, it shows all the phalloides in with them too. And that's because in many of the herbarium records are uncertain. But the dots on the map is mostly oceanic maritime influence. So the east coast of Europe, inland into areas of the Ukraine. You unfortunately have the Baltic Sea on the east and the Black Sea, and then you have all the dots on the map that I could see were sort of within range of some maritime influence that would break. But when you get into the Eurasian steppes, and there aren't many dots there, but then there maybe aren't many observers too. But there's the odd dot that's really getting into that real continental sort of thing. But I don't know if it's the citrina or the phalloides. But so it's and this is we now have generating maps based on the herbarium specimens that we have. And we're also trying to figure out how to get INAT observations that we can trust onto here. But it's been a tough thing, Mushroom Observer and INAT. It's very hard to, we're really hesitant to try to dot on maps based on those sorts of records. So you can see that there are real clusters in the urban areas. And this is Vancouver now with, yeah.
To see how far their mycorrhizal will go, like up the tree. So it goes much.
Tree roots can go a long ways, so you can have mushrooms popping up really quite a distance away from the host tree, especially where you have things like big open lawns and the roots can go quite a ways. And that's why it's really tough. We ask people to note trees that mushrooms are growing under or nearby. Some people think it has to be right up against the trunk to count, but it can actually be quite a ways away. And that makes things a real problem when you get mixed trees and you get a mushroom sitting somewhere and you don't know which tree they're connected to. And there was one case where we found phalloides growing with horse chestnut, which isn't a really well-known ectomycorrhizal host. But the location, there were oaks around the corner and there were different trees here and there. But I was sort of looking at the infrastructure, like where the roads are and barriers that might interrupt tree root growth. And we're pretty sure that this one location, it's the horse chestnut that they are associated with. But in some cases, like one person found them growing apparently under a plum tree. And she was all excited. She said, I found them under a plum tree. So I went and visited the site and I looked at it and I said, well, I'll look at it. Like, Vivian, you gotta look at that. There's a hornbeam tree over there, and I pasted out for her. And, and then I also noticed, also if you notice, there's a stump there where there used to be a tree. So it could be that there was a host tree that was cut down too. But anyway, that's, I think that's it. Yeah, okay, so these are some of the people involved in the survey. This is just a small, several people in our club were decided to dedicate some time to this, and they adopted mass plantings and walked the streets every couple of weeks for a couple of seasons. And they'd photograph the mushrooms, write out a little thing. I tried to get the specimens to myself, or Vivian Mao was the one that sort of designed a lot of this, and so we try to dry out the specimen and put it in the UBC herbarium. And so we got dots on maps, and that's it. And it's an example of a citizen science project that has, I think, contributed good stuff. And I would encourage you to think of something like that for your club, and needn't be Deathcap mushrooms. Just city street trees are a fascinating way to set up a study because you can get information on the trees. They're often mass planted. They're like little study plots laid out for you. They're in your neighborhood. If it's right in your neighborhood, you can adopt a street nearby that has 5 or 6 trees of some species and just follow them around the years and see which mushrooms come and go. And it could have implications for tree health for the city parks board, or, it could be, anyway, so that's a project. Right.
Thank you.
Thank you, Paul.
All right, okay. Yeah. Great.